Hey, tonight it's my pleasure to introduce to you Ingrid Jordon-Thaden. She's with the Department of Botany. She just came here in November to be the director of the Botany Garden and Greenhouse. She was born in Omaha, Nebraska, and she went to Omaha Vincent High School. And then she went down the road about 72 miles to get University of Nebraska at Lincoln, and she got her bachelor's degree both in chemistry and in horticulture. And she stayed at Lincoln to get her master's in biology. And she got her PhD at the University of Heidelberg in Germany, and that PhD was in biology. She postdoced at the University of Florida in Gainesville, also working on living plants, but then she went to Berkeley and worked on dead stuff. In the herbarium. I was looking at all this stuff, and all of a sudden she's working with dead stuff. But then she went to Bucknell University and went back to working with live stuff. Went back to Berkeley for a while, and then came here, like I said, in November of 2017. As a botany person, I think, which is what I was a long time ago, it's an amazing thing to walk through her greenhouse. It's a splendid thing to walk through her garden, to see the world at your feet, and plants from every region right there at the bench where you can check them out. And it's also fun because of the solid layer exposure that the garden has. It's one of the earliest places that the dandelions bloom. Sorry about that.
Oh, we don't have any dandelions.
So, the other thing that she did, which is great, she's going to talk about my three favorite things, which are the three missions of our beloved Public Land Grant Research University. Our missions are research, teaching, and outreach. Please join me in welcoming Ingrid Jordon-Thaden to the Wednesday Night Lab.
Well, thank you for that really fun introduction. That was pretty awesome. I do have to say that I worked with dead things many times. So herbaria have been part of my life since I started studying plants a long time ago. So I love being right across the atrium from the herbarium because at least I'm close to the dead things. So that's good. So I would like to give you a little bit of stories about how I got here. Being director of the garden and greenhouse kind of means I'm got a lot of different things in my bag to pull out and use, and so it's good for you guys to hear like a little bit more of my stories. And so he mentioned to you that I did my bachelor's degrees in Lincoln, where I created my own botany degree, what was a combination between chemistry, biochemistry, chemistry itself, or horticulture, range science, and essentially worked in all these different, see, it always happens, right when you start, it stops working. Yep, okay, so I worked in chemical taxonomy, was my ultimate senior thesis, and really loved talking about evolution of plants, in particular chemicals in plants. And so during my master's, I got into proteins in plants, and I was working in corn mitochondria, and I was really lucky at the time because I got to fall in love with botany even more by being the director of the undergraduate labs for botany. And then we worked, me and two other of my colleagues, my master's advisor and one of my good friends, rewrote this manual here, both for anatomy and for general botany, and they still use it today. I think this is one reason why I got this job, actually. And then after doing American academia, I thought, let's do something different. And my husband and I, my husband there's right there, my daughter right there, we decided to do something completely different, and we went to Europe. And he joined the Max Planck Institute, and I joined the University of Heidelberg, and did a PhD solely there, not connected here, but all the way there. And while I was there, I worked with this particular duo, Professor Marcus Koch and Ihsan Al-Shabazz. He was a professor at University of Heidelberg, and Ihsan is a professor at Missouri Botanic Garden, and so they put me in love. They turned me towards the love of draba, which is this cute little mustard right here, which I could give lots of talks about draba, which I won't. I'll just tell you a couple cool things. So while I was doing a lot of lab work, being that I'm basically a chemist, I'm a plant chemist, I also was very big into growing plants all the time. And so Marcus could tell that I really, really wanted to grow plants. And so he said, hey, I've got this freezer full of seeds, of lots of draba seeds. Does that interest you? Because I jumped at that immediately. And so I grew about 35 different taxa, about 600 plants, and then just started looking at all of their different modes of reproduction. So plants can do a lot of different types of reproducing. They can be compatible to themselves, so they can self-pollinate. They can be self-incompatible, so it makes it they can't actually pollinate themselves. They can self-fertilize, or they are insect dependent, so they need an insect to do their crossing, or they can also be apomictic. And this is all found just in one genus, which is actually kind of rare to have that many different reproductive modes in one genus. And so that was going on in the background for 4 years. I was doing phylogenetics in the lab and with dead things, mostly. All of this was about 1,500 different herbarium specimen pieces, which I had inherited from other students who had gone to other herbaria and collected material. But ultimately, I came up with a phylogeny that was published, and you can read it right there. Lots of good things about it, but it can be generally grouped into 3 main clades: Europe to Iran, then this whole huge circumpolar region, also into Asian mountains, and then down into the North American and South American Cordilleran groups. And so since then, that was published, the last one was published 2013, and since then I've been doing more molecular work for this with new markers and getting into all those new fancy machines and new sequencing technologies. And so I have other projects that are almost finished in this particular realm to redo that actually already. And so one thing about my greenhouse work that was really exciting is I noticed that when I read about draba and different treatments of draba, so if you know about what a monograph is for a genus, so it will have descriptions of all the different species, and usually it's a very long book, 200 pages. And in the 30s, a German botanist, Schultz, he spent a lot of time looking at draba and also lots of mustards, but of course, like all good mustard taxonomists, they fall in love with drabas. He grew them, and I could tell he grew them because he referred to plants and characters which happen to have different characters over time, and he would say after the plant is 3 to 5 years old, then it does this. And so I said, well, okay, I'm going to have to grow them. And so that was one of the reasons why I started growing them too. And so this particular plant, the one that grows or creates seeds without sex, I thought that was pretty exciting. And there was another botanist who thought that was also really exciting at University of Ottawa. His name was Mulligan, still there. And he had written in one of his publications that it's possible that there might be sexual plants of this particular species in Montana and Wyoming. And so I said, oh, that's really cool. I think I'm going to travel all over Montana and Wyoming and find this sexual plant out of all of them. So much to my family's confusion, I decided to spend the next 5 summers taking field trips to collect this particular species, 2 to 3 to 4 weeks at a time, from the Yukon down to the Sierras and then over to the Medicine Bow, which is in the far eastern Wyoming. And after a ton of work, which I could give another talk on if you're interested in, we essentially found out that about 3% of the time, the seeds will have been produced sexually. And 97% of the time, it's asexual. And it's not just in Montana and Wyoming. They're all over that you have these occasional sexual seeds. And then it formed these 3 individual clades. Here you can sort of see the third one is this small little clade C, which gives us an interesting phylogenetic and phylogeographic story, which I won't share with you now. Oh, I'm going to kick on the wall there. So that's draba in, what was that, very short period of time. And next I'm going to talk briefly about Tragopogon, which is called goat's beard, and it's similar to, in growth form, to chicory, if you're familiar with that. I'm not sure what the Wisconsin native name is for goat's beard, but It's goat's beard? Okay. Yeah. I don't know all the common names here yet, so you have to fill me in. I was really lucky to join this group at University of Florida, Pam and Doug Soltis in Gainesville, and they had been studying this particular hybridization event that had occurred in the 1950s. They started studying in the 1980s on it, and in eastern Washington. And it's been a really unusual and special case of hybridization where three parents, I'm going to have to walk over here, sorry, because I don't have a pointer. These three parents in the corner, those three are introduced from Europe with weed seed from wheat bags into the Palos Prairie in eastern Washington. And naturally, these two hybrids in the middle, the T. mycellus and the T. murus, they formed naturally in this new habitat where these three parents kind of showed up in this special, different style of habitat that they didn't have back in Europe. And so it was observed by Onaby, who was a botanist in eastern Washington, and he said, oh, there's a new species, really exciting, new hybrid. And it turned out it also had double the amount of chromosomes, which is a special kind of hybridization called allopolyploidization. And so it will continue to have both parental genomes, but then form a new phenology and everything completely different from its parents. And so I spent a good 6 to 8 months solidly in the lab generating these crazy trees, which you see over here. This is a genotyping tree with many different tips and individuals in the study to basically show a lot of different things that happened in the genome when you become a polyploid. And one of the things that happens is that the genes will actually start to slowly be lost, and they call that diploidization. So once it had the double genome, and then it just goes back down to smaller. And we had thought it was a very slow process, like possibly hundreds of generations. But with the study that I finished there, we found that it actually starts happening immediately upon the first generation. And I can explain more about that later in another talk if you want to invite me back.
Okay.
And then once we collected those leaves and did all that genetic work, they were only about that tall. And I said, well, I'm not throwing these away. That's 900 plants that we could do something else with. And so Pam Soltis agreed to let me grow them out into wonderful, beautiful greenhouses in their horticulture department. And I proceeded to measure every other phenotype that I could imagine, like growth rate and so on. And now I'm correlating those phenotypes with the gene activity. And in general, most of these characters, which I recorded, showed that the hybrid was winning over the parents, which is what we sometimes expect. And also in the native habitat of this particular eastern Washington, the parents are declining, but the hybrid is going up. Okay, so then the last little postdoc project that I got to be involved in is with Professor Chris Martin in central Pennsylvania out of liberal arts college called Bucknell University. I have to say that because I'm not an East Coaster, I did not know about this university till I met Dr. Martin at a conference. But it's a great institution, and I welcome anyone to learn more about them. So I was in the biology department there with a wonderful team, had 2 beautiful greenhouses to grow giant plants with. They were 5 to 6 feet tall, woody plants. This is related to the eggplant, mostly related to the eggplant and also the tomato. And we studied sexual systems within this particular group of Solanum, and they were all from Northwest Territory, Australia. And he just got back again from Australia. He's been studying these particular plants since about 2006, or 5, actually. And ultimately what we were able to find out about while I was there was that there were a couple of really cool things about their pollen. So these plants will be what's called dioecious, some of them, which means that they'll have the female plants on one house and the male plants in the other house. And so they have to get together in order to have a new baby, right? So the theory is that these plants that separate their sexes on different individuals would have higher genetic diversity because it sort of forces outcrossing. And we found that, yes, they do have higher genetic diversity because we were able to compare different Solanum species within the same area that were hermaphroditic, like most flowers are. And the hermaphrodites had lower diversity, genetic diversity. And then also these dioecious plants, they look dioecious on the outside, but just like the draba, being that it's kind of weird and sometimes it's sexual and sometimes it's not, it also makes pollen. The female does, even though it can't use it. And so I've gotten kind of into this whole process of making sex parts that you don't use. Seems kind of like a waste of time. So there's, these pollen in the females will produce pollen. It looks completely normal from all stages of development, and at the very last stage, it lays down this bad, ugly-looking coating. And if you've ever seen SEM pictures of pollen, they're really beautiful. They have these sharp, pointy things, and they're perfectly geometrical, and they're species-specific. And this particular female that doesn't have active pollen but produces it, it's like a bad spackle job. It's like somebody just kind of went, and just looks terrible, and all the pores are covered. And so the pollen is essentially inactive at that point because it can't germinate. Even though it has the full 2 cells inside and it has the sperm, goes through all that process, and then it can't lay down the outer layer, which is called the exine, properly. So those are 2 papers that I'm working on to get those out. And that's a little bit of background about what I did before I came here. So I'm going to switch gears and tell you more about what I'm doing here and why I'm here. That is a really dark slide. Essentially what that says is that I'm planning on maximizing our research space and our teaching space in the greenhouse, and if you ever come by, you'll see maybe sometimes it's really crowded, but there's some stuff sitting around, you know. So I'm working on organizing things. And then also a major project that we've been doing is creating and trying to start a new curation database, which has been a little bit lacking for a while, and so we're trying to bring it back up to speed and try to adopt some new software, bring in some more public activity to the curation. And then we want to elevate the visibility of our collection in the greenhouse and gardens because not enough people know about it. It's not in the tour books. It's not in the hotels in Madison. There's there's nothing about our garden and greenhouse that is really publicly known. And everybody I talk to say, where are you again? Allen Centennial? No. Olbrich? No. And I'm like nobody knows. And so I'm trying to make sure that we are known. Right? So that's like my number one priority, I suppose. So I'm going to talk to you about some of the research that is sort of a little bit finished recently since I started and stuff that's starting up in the next months. And one of the projects that started almost immediately when I arrived is one particular drought-resistant project of trying to understand how different species are either more or less tolerant to drought stress. This was done by Chris Krieg, who is a graduate student of Emily Sessa, who's at University of Florida, who was a graduate student here, and I'm not sure when she left. When? 2000.
5 years ago.
Yeah, 5 years ago. And so she's coordinating with Professor McCullough, who's at the Botany Department, and they're looking at these 2 particular ferns, and it turns out the top one there has the ability to handle drought stress much better than the bottom one. And you can basically say that that's minus 162 means it's more drought tolerant. I won't go into more details, but they don't really know what this means yet, but it's a bigger study that they're working on with other species too. The Medea lab, Hiroshi Medea in our department, is also working quite heavily with the garden and greenhouse. He works on the biochemistry of a pathway in the plants that's called the shikimate and amino acid pathways. And this is basically the pathway that gives rise to different types of chemicals in the plant that give the plant color. In particular, betalains, which are found mostly in the Carophyllales. The Carophyllales, this word right here, this is a group of plants that's been collectively grouped for a very long time and actually has pretty successfully survived the phylogenetic untangling with all the genetic work that's been going on for 20 years. And what Hiroshi's big recent study showed was that the evolution of the gene that is involved in developing the backbone of betalain evolved before the actual betalain itself, which is interesting. And so all the different families on this list, you can see that all the empty circles, there's a lot of unknown information. And so he recently asked me to help support him in a new grant that would then bring together more screening of more plants in our collection. Another cool project that has come into our lab, or into the greenhouse, that they're taking our plants into the lab, is with the Department of Bacteriology and Agronomy, Professor Jean-Michel Anna and his postdoc Vanya Panchkiev. He's Russian, and I'm very bad at, sorry. And they are interested particularly in bacteria that fixes nitrogen, which is usually found in beans and so on in the root nodules. But it's been reported to also be found in this picture doesn't quite do it justice, but you see this slimy material here around that. So that's called a mucilage that's being exuded directly from those aerial roots. And this picture here is corn in particular. And so turns out to be nitrogen fixation bacteria in that mucilage. And so they're looking to see whether there are other aerial roots in the plant kingdom that also have this cyanobacteria, like our orchids and arum lilies. And the tricky thing is this exudate only comes out about 20 to 40 minutes after you water the plant. And so we're texting, oh, hey, there's exudate, So they'll come out. So Vanya comes out with her little vials and collects and goes and does a test for the nitrogen fixation. And then the Anne Pringle lab in the Botany Department, she has a new project which she's put a graduate student, Nora, and an undergrad, John Zuber, onto another cool project that has to do with the microbiome of the soil, particularly involved around trees. And so we have in our greenhouse some seedlings of oaks and pines and maples, and they're looking at different types of inoculants into the soil. Native inoculants from native tree soil, like at the base of native trees, and also ones that you would buy at the store, essentially to see if these ones that you buy at the store are actually introducing invasive inoculants or dangerous inoculants, or they're not doing anything at all, and it's a waste of money. So that'll be a cool project to see how that comes out. In our greenhouse, they're just doing the pilot study to test the methods, but they're going to be doing thousands of plants at the Walnut Street greenhouse. And then another rather large project that we are involved in now is growing cotton. This is the first time I get to grow cotton. I'm kind of excited. I was expecting corn to come to my life really quickly because I've grown corn multiple times, but cotton is now here. And the Gilroy Lab recently was funded through Target Corporation in the Cotton Sustainability Challenge. And together with Dr. Swanson and Dr. Barker, they have a particular genetically modified cotton, which is on the far left of my side or the right of your side. And you can see those roots there are more fibrous. And those fibrous roots then have a better ability to absorb water and nutrients, which makes them stress tolerant and also salt tolerant. So what we are going to be doing is taking those genetically modified cotton, growing up a large quantity of them to seed so that they can be sent off on the space to the space station. And then they'll be sent up there to do studies with the salt tolerant and drought tolerant in space. So that's really cool. And then one of our, I would say, classic uses of a systematics collection is that we are part of a larger Lamiaceae and Lamiales study, in particular one genus that just got an extra bit of funding called Salvia in the Lamiaceae, which is the mints. One of the undergrads that was working as a assistant in the greenhouse is now also working in Professor Seismith's lab, who's in our department, on this particular genus. And so she's going to be doing classic systematics and different kinds of measurements of the morphology of the flowers, because they have a very distinct type of flowers, leaf, trichomes, and so on, and then bringing those leaf tissues to kill them and put them through the DNA process and doing phylogenetics as well. So that's just a small bit of research which is done in our collection, and there isn't any research currently in the garden. So if you know somebody who wants to do research in the garden, tell them to come talk to me. All right, but one of our main goals in the greenhouse and the garden is for teaching. We have, I think, approximately, I counted 18 different classes that use our collection at one point in time, either in the fall, the spring, every other year, or the summer. And so we are offering the collection to the students and the professors to use for their classes and also for study on their own. And so this particular, it looks so much better in person. That picture really doesn't look, yeah. Anyway, this is our beautiful desert house, and it's one of the houses which we are able to divide up quite nicely by family. For example, right there in the middle of the room is the Cactaceae, while on some of the other sides of the house you have some of the other families. Like on this side is the Euphorbiaceae, and on the back is mostly Asparagaceae and so on. And they are all in pots so that we can bring them upstairs to the classrooms if needed. People ask me that every time they come in. They're like, why are they all in so many pots? I'm like, because they're mobile. They're mobile plants. They move around the building. It's really cool. Another thing that we have in our greenhouse is a pretty significant carnivorous plant collection, and that is due probably mainly to Professor Givnish, who's here in the office, or in this office that we're here together, in this room. And one particular famous carnivorous plant that we have is a nice collection of Venus flytrap. This particular species is endemic to the North and South Carolina mountains and bogs, and it is officially on the list to be officially considered to be officially endangered. And University of Wisconsin-Madison's Botany Department was instrumental in getting it there, in particular Dr. Don Waller. We also have a relatively large orchid collection, not as large as Ulbricht, but we do have a good collection, which we're working with a couple of the other orchidophiles, particularly Professor Ken Cameron, who's the department chair, who studies orchids, particularly vanilla orchids, which we have one blooming right now, and then a couple of his graduate students who are very interested in maintaining and helping to make this orchid collection a good outreach opportunity. And then another thing that's different about our collection compared to many other greenhouses that I've been to, I actually had a running list of all the botanic gardens and greenhouses I had been to for many years, and I looked for it to count and I couldn't find it. So I was trying to remember them all and I couldn't, but it's definitely more than like 3 dozen. And I would say that we have one of the largest aquatic collections that I've seen inside a greenhouse. Probably also to Professor Givnish's thanks that this is here, but this is a particular bench that we have that has about 3 or 4 inches of water all the time, and then we also have a 700-gallon tank in the middle of this room that has koi in it, which we moved a few out to the pond yesterday. And then you can see on that little white bench over there, there's some more underground, like fully submerged and floating aquatics. And we have a very good representative of many species of aquatic plants. And then we also have a very large fern collection. Again, due to Professor Kimish's work. I know some of them are yours. It's okay. You don't be so humble. But anyway, we have a very good representation of fern genera, which is really important because the identification characteristics that you need to learn ferns, you really need to have a wide variety of the fern genera. There are many, many more species than what we have, but we have a good representation of the different genera. Our moss collection is tiny, but cute. We could definitely use some more mosses and maybe an improved moss growing situation so that we can induce sporulations, which is where they produce their spores and look really different. And then I just want to give a plug for our beautiful aquarium that we have upstairs in the atrium of Burge Hall, which was almost 100% maybe funded by Mr. John Glaser, which some of you may know, who used to be at the Wisconsin Public Radio for many years. He has maintained this particular tank, which is about 120 gallons, and another one at Nolan Hall. And we have a student who helps us with this, actually the one that's working on the tree microbiome. And it's a beautiful tank, and we get to take care of that as well. And we have 5 other tanks down in the greenhouse, which I've added since I kind of resurrected since I started. I'm kind of a aquascaping junkie, so this works out pretty good for me. All right, so I'm going to briefly talk about the garden. The garden this is a picture that I found someone had posted. Someone in the room, and I don't know where they got it actually. It's approximately 2006, shortly after the garden was redone completely. You can see those pathways and sidewalks that wasn't there, but in 2004 it was completely remodeled and added on to be the structure that we have today, which was actually the landscape design student project, which was a great use of campus resources. And it is one of the first gardens organized by the phylogeny of angiosperms in a particular group called the Angiosperm Phylogeny Group 2 was when it first was laid out. There's APG 1, 2, 3, and 4. We're on 4 now, and that has to do with the development of our understanding of evolutionary relationships between plants. So there were some changes that were made to bring it up to APG 3, and so now I have to bring it up to APG 4. So I did my first step as I changed a few of the labels on the map, which you'll be able to see when you go visit. But one of the things I'd like to be able to do is increase the signage throughout the garden so that more information about the different beds is available. On the far east side, where the number 2 is, it's mostly monocots, and although the number 2 is actually grasses, grass, fescue, blue stem. And then on this side is everything is non-monocots. And then evergreens and other gymnosperms and ferns and things are scattered throughout. We also have a notable tree, the Newton apple tree, which was planted in 2001. And this particular tree is thought to be descended from the actual stock of the apple tree, which hit Newton on the head when he came up with his theory of gravity. I had the idea of, well, let's do a DNA test and let's get some. And Dr. Gilroy was like, I don't think we really want to know. No, let's not do that. It's good. It's good. Let's leave it. So we're just going to leave it. The other thing that we're doing in the garden is we're going organic. I also could say we're going organic in the greenhouse as well. We've cut down, Kara Streekstra and I have cut down drastically on the spraying inside the greenhouse and have 100% eliminated spraying pesticides in the garden because we decided it's not necessary, really. We don't really need to worry about it so much. And people use the garden all the time, and they're sitting on the grass, they're laying on the grass, they're eating their lunch. children are playing with no shoes on. I just said, you know, why? We don't need to do that. So we got rid of it all, and so you're just going to have to get used to the clover and the dandelions. But, you know, the dandelions get eaten so fast that it's not that bad. But you can see the clover there. We've introduced some other bee houses and some various other types of fungi that are good for controlling common lawn pests or lawn fungi. So be patient with us, but we're going to see how it works. We also have recognition stones or recognition pavers that we have in the plaza that was put in 2004, and so we're still getting requests for those, and we place them down in the middle if you donate over a certain value. And we're adding irrigation to the garden slowly at first. We're going to try just using soaker hoses. You can see here, this is my staff laying down some soaker hose. And so we're interweaving it throughout the annuals, and then we'll cover it with the mulch, and that'll help with water loss. So we'll put instead of using a sprinkler on top, which is what we've been doing before. And then also the garden has a small amount of art and architecture to look at. So we have a triptych of a sculpture art piece that's found in the garden. You can see that as well. This one is one of the ones that refers to the flower. And then we have a beautiful gazebo with cedar plank ceiling and a couple of other structures, some different arbors and so on that are nice to look at. But one thing I'd like to do with one of my new new hires that I've just brought in. She's very interested in public gardens and public green space, and so she wants to help bring in some more ideas of art projects and other activities where people can participate with the structures in the garden area. So that leads me into the outreach projects which I have going on. Some of them I've started already, some of them I'm just getting oriented with. One of them is that I thought we definitely need to get the Botany Club more involved in the garden and greenhouse. We have about 30 students this year. I understand that our president just graduated, so we're going to have to recruit and make sure that that Botany Club stays strong. But I brought them down to the greenhouse and gave them a lesson on propagation, and they made little houseplants and they had a great time. And it reminded me of a statement which someone had told me a long time ago when I was a student, that it was really great that I knew both botany and horticulture, because a horticultural or a botanist knows the plants and a horticulturalist knows which part goes in the ground. And so I think that the botanists, they benefit from this because they don't actually have plant propagation as part of their schedule. They have to take that extra, right? So. Another project that landed on my doorstep was a student that came in part of the Climate Reality Project. She came in asking if they could grow plants in our greenhouse and use them for a sale, and that worked out great. They made like $300 for their club, so I'm hoping to do more of that, and we gave them a space like this big. It was not that hard, so it worked out perfect. And then I'm also looking to get involved with the Madison Area Technical College. They actually have a botany program there, and there are quite a few students looking for experience and volunteer work. One very small niche program that they have is they have a placement program for students with intellectual disabilities. And so we have our first student who's helping us and doing really great and part of the whole city program. And then if we improve our ability to control the summer heat, which I'll talk about at last step, I would like to become a plant rescue center for the U.S. Forest Service and Fisheries and Wildlife. At the borders, plants are often collected because they were confiscated because people collected plants illegally. And usually those plants are special. There's some reason they brought them over. They didn't just pick something fun that they saw in the corner and bring it home they're usually actually poaching those plants and bringing them in. And so those plants then need a home because they can't stay at the office at the border. They have to actually have a place to grow so they don't die, because technically they still belong to the country, the origin that they were taken from. And so the U.S. Forest and Wildlife Service, they have a program where you register to be a plant rescue center, and they will send you plants if you have space for the particular area that you have that was confiscated. And you just have to promise to mail it back to the country if they want it. Often they don't, but it's good to be able to know that you're saving endangered plants, because one of the eye-opening experiences I had in Heidelberg was our Botanic Garden there had plants that were completely extinct in the wild in Africa, and it's not because of collection at that particular time, because that was back in the 30s and 40s when the Germans were collecting plants and bringing back to their greenhouses. It's mainly because of habitat loss for those particular plants. Another group that I'd like to join, and other gardens in town are also part of this group. This is a global group called the Botanic Gardens Conservation International, and it basically means you're going to work together with other botanic gardens to safeguard plant biodiversity globally. I don't really know what our role will be there, but it's good to be on the list in case people need assistance or we can help out in any way, and also just contribute to the efforts that they do globally. And then because I'm still a geneticist, I really want to stay involved in the genome world, and this is something else that we can do as the biodiversity repository that we are. It's called the Global Genome Biodiversity Network. And here we would basically take individual tissues from all of our individual living collections and then store them appropriately, either frozen or on silica, so that they could be maintained for discovery, other research that would happen in another part of the world. And so you become part of this network that you are known as a facility that has certain genetic resources. One of the first steps I'm doing in this is we're actually bringing in a very important flowering plant called Amberella from Pennsylvania. And another outreach program that I just started getting involved with this week. I have 2 interns this summer, and they are high school students in the diverse Madison area who are interested in media and information technology. And so it's part of the Do It program. And they sponsor and actually pay these students. It's their first job, and they come in and help you with anything that you have to do with some sort of media-related activity. And so my first intern that started on Monday, she's helping me improve our website. And she was to make a YouTube video and get me on Instagram. She's like, she's really cool. And then another really successful program through NSF is called Planting Science, and this is targeted mostly to middle schools, but it is also reached in some lower and high schools and some even elementary schools, and it partners with teachers of biology who want to learn more about plants, and so you become an expert of plants and you get connected to a teacher. And so I've been part of this a couple of times, and so now that I'm here as the director of the Garden and Greenhouse, I'd like to get involved a little bit more seriously. So this is an NSF funded project that's been going on for, I'm actually not sure how long, but I think it just got funded for another 5 years, I believe. And then lastly, I'd like to make a Botany Workshop. This is the grandiose idea that I've been talking about to different people, where we would bring in experts of all of our botanical community, which we have many of here in the Greater Lake area, and teach some hardcore botany. And we teach it right out of our greenhouse and our Burge Hall, but they wouldn't be students. So these would be 1 to 2 day workshops where you would learn all there is to know and just cram your head full and walk out sort of like numb. That's the kind of workshop that we're looking for. All right, and so while we're doing that, then it's important to do some interior scaping inside our greenhouses, so there's some improvements we want to make it look a little bit more appealing. This is one of the favorite rooms of our students from all over campus. They come and sit here. So there's some improvements that can be made. One in particular, which would be a living wall for this room. Here's 2 examples of living walls. The one on the left here is one of the largest interior living walls from the San Francisco Museum of Modern Art. And then the one on the right is a smaller example where it surrounds 2 doors. And what a living wall really is actually an art installation with plants, not a horticultural installation as much. It is, but it's more about the art. And so you can see here, this is like a picture color by number, but with plants. So you'd put the different species by the different colors, and this is what the San Francisco living wall is. So here's my first draft, which I have to say is quite embarrassing, of the living wall that I'd like to create. But it would be in our larger room where we have a lot of people who come to sit, and then be right next to the door to the classroom where we would have workshops. And so I'm working with some art-inclined and curatorial people on campus to work on some grants to bring some money in to put something like this in, because it's definitely not the most cheap way to handle plants, but it is quite stunning, and it is we could be doing it in a very exciting way with epiphytes and ferns and mosses and all kinds of cool things. So you're probably wondering, how am I going to do all these things? One thing I have to do is I have to fix some things. You know, the greenhouses, they are like living organisms. They breathe, they sweat, they break, they leak, right? And so there are things that we need to fix. And so in 2006, the exterior part of the greenhouse was replaced. There's 8,000 square feet of the greenhouse. And tempered glass was put in, and it's still in great shape. But unfortunately, the funding for the inside of the greenhouse wasn't quite enough, and so there are some things inside which make things really difficult, especially the heat in the summer. So we've, with our own special media specialist, we created Sarah Friedrich. She created a mailer, which we're getting ready to do a 6-month fundraiser. Some of you may get them in the mail, probably, and they'll be sent out very soon. And with our sort of main goal is to improve the climate zones in our greenhouse, because we have all these different diverse plants, and they're doing well most of the time, except in the summer. The summers are longer and hotter and more humid, and we just don't have proper controls in order to keep them really happy all summer. So in the fall, we end up playing doctor and repairing and removing dead plants and bringing plants back to life. And, you know, it would be great if we could have good controls 12 months of the year. So these are the 12 different rooms. You can't read those names. And ideally, I have cool and dry, like alpine, warm and very wet, warm and wet, you know, cool and moist, all these different ratios of cool, of moisture and temperature in order to grow the different species. And also be able to close the doors, because right now we have the doors are all open most of the time. Not all of them, but many of them. Because otherwise it just turns into a really, like, you know, an inferno. And so in the summertime, this is more what it actually is, warm and moist and very wet. And so that's not great for a lot of plants, especially when you're trying to grow a diverse array, and people have carried plants with permits very carefully back from different countries to bring into our greenhouse for teaching or for research, and we have to be really careful not to kill them. And we're worried about some of our plants not being able to survive. In fact, we have a baobab collection that's kind of like on the brink of not being able to handle the extreme moisture that we have in there. And so one of our first steps that we did was we twisted the arm of the LNS, and they were super great, and they let us buy new shade cloth, which you can see here, and it's now, up since last week, and it really does help. It doesn't fix the problem, but it does help a lot. And so we'll see how it does this weekend when it gets up to 92. But I'm hopeful. I'm hopeful. We also were awarded another pot of money through our CapEx exercise to put in irrigation benches. So we are putting 5 in areas where we need them the most. And we plan on Also improving some of our lighting structures. You can see here the plants are growing above the lighting structure, but we don't have the ability to raise the lights as much. So there are modern ways that we can upgrade this and also convert all of our hot fluorescent bulbs to a cooler LED lighting, so that might help with the heat. So lots of different things that we can do. We could also adopt a type of electronic controls. We have electronic controls in one of our greenhouses, Greenhouse 1, which was redone in 2007, but the other houses don't have the electronic controls, and this would greatly help out. One of the cool things that a lot of our students like is when we put together habitats of plants that they would see in the natural ecosystem. Like here we have a bog that's sort of constructed out of rocks and logs and things, and it's really popular. But some of our other plants, like the Mediterranean, subalpine, and alpine plants, are crammed onto these tables, and they don't have any height or ability to be displayed in a way that they need to. And also, these particular plants might do better if we could move them outside into some cold, or if they could really get colder. So there's a lot of, they're just doing what they can, and the ones that are there are the ones that have survived our heat. Because I've studied draba, I'm really into alpine plants, right? And so alpine plants require that you be able to control the humidity, because you really have to have low humidity to grow alpine plants, or they just rot. This is my garden at home, which I'll see how it does. I joined the Rock Garden Club in town, and so I'm really excited. And we went to Flower Factory, and I spent $100, and I was like, really exciting. So about 8 of those are drabas. So I'd like to be able to do more of that in the greenhouse. And in the succulent house, there were built these larger raised beds, which would be similar to what we could do for the alpine plants. Those raised beds were put in quite a long time ago. I'm heard it's about 1985 or so. And they've lasted a long time. And so that pretty much sums up what I wanted to tell you today. I want to give a couple of acknowledgments to some very important people who've helped me in the last few months. One is the Curatorial Manager, Kara Streekstra, and she has been here for about 7 years and did her degree here as well. So she's our institutional knowledge for the collection and for all the facilities. And it's been really great working with her. I'm really excited that that we're working together for a long time, I think. And then I also want to thank Mo Fayez, who many of you may know, who was the director of the Garden and Greenhouses for 33 years. He comes in still every Wednesday and works in the herbarium and offers advice and help, and is always there by email, and it's been really helpful. And I also want to thank the Botany Department for everything that they've done to get me settled, and everyone has been so helpful. In particular, Dr. Gibnish has really bent over backwards to help out. To make sure that things work out the way that I was hoping them to. And I really am thankful that they've trusted me with their collection. So if you have any questions for me, this is yesterday in the pond. I was organizing plants and releasing fish. I'm able to answer any questions, and then we could go for a little walk.
Greenhouses do you have, and how many different locations around campus?
Oh, so the Botany Greenhouse is just centered right at Burge Hall, and we have 8 greenhouses, and each but 12 different rooms. And so the other greenhouses on campus are run by different departments. Yeah.
How do you answer the why should me, my neighbors, Joe down the street, why should any of those people care about the Botany Garden and Greenhouse?
Oh, why should they care about the Botany Garden and Greenhouse? I would say they should care about the educational opportunity that the Botany Garden and Greenhouse has for them, not only for possible students who would be taking classes there, but also for the community that comes in and learns about botany and relationships between plants and how plants live and breathe and die and all the things that plants do. Botany is very different from agronomy and horticulture. They have completely different goals. So botany is very much about learning about how the plant functions and how many plants there are. Like, we are really focused on the diversity, the physiology, the ecology, and without these fundamental questions that we answer in botany, you couldn't apply them in agronomy or horticulture to real, like, economic situations. Yeah. Yes.
2 quick points. First of all, the plant you mentioned, Amarella,
Yes.
which you're picking up in Pennsylvania, I just want to make sure everyone understands it, doesn't grow naturally there. It grows naturally in New Caledonia,
yes.
in the middle of the Pacific, actually about 1,500 miles from the shores of Australia. And it is sister to all other flowering plants. So it diverged from their common ancestor about 135 million years ago. And secondly, we're delighted to have you aboard.
Yeah, Ambarella is a really special plant in that it's considered what's called the most realistic first flower that we can see today, the extant plant that has a flower that looks like the ancestral first flowering plant. So it's pretty exciting, and its genome was sequenced, and I've been around an Ambarella plant in Florida while I was at the University of Florida, because they did a lot of work on that particular species there on the chromosomes of that species. And that stock was actually propagated and then passed around through different greenhouses across the country. And so we're going to be getting some of these special individuals which are part of this special genotype that was first sequenced. So it's kind of exciting. Yes? Oh, yep. Go first, yeah.
We could sing about amaryllis written by geese. It sounds apples to centipede.
Do I have to repeat that? Because I'm not sure. Do we have amaryllis? Yes, we do. We do. But I guarantee that Ken Cameron, our chair, has more amaryllis at home. Yes?
As a person who was working in some greenhouses, what did you think when you came and saw the 2 trees in the southern exposure of your greenhouse?
Oh, yeah. The question was asking about the 2 trees on the southern exposure of the greenhouse when I first arrived. I said, well, it's not that bad because there's a head house that's a bigger problem, actually, because it's on the southern side. But, you know, it's actually, we don't get a lot of shade from those because of the head house. If we moved the head house and changed the organization, then I would, you know, talk about those trees. But they're okay where they are. Yeah.
And you have in the greenhouse plants unique to Wisconsin, such as, like you pointed out, Venus flytrap. Well, they exist in Wisconsin too. And I don't know if they're the same, how many different species of carnivorous plants we have. So it would be nice if we had something that was unique in Wisconsin or in the upper Midwest.
Yeah, so the question was whether or not there are plants unique to Wisconsin in our collection. And I would have to say, not really. Most of the plants we have are characteristically important for their teaching capacity. We don't really have a lot of native plants. Even though we do have plants that do occur in Wisconsin and the Greater Lakes area, but we don't really have a designated area with just native plants. Occasionally there are some native plants in there, but it's not necessarily our primary goal. Yeah. We may have more native plants in the garden, actually. Any other questions?
If not, why don't we adjourn, and if you'd like to leave them, I'll lock up and join you, but if you'd like to leave people either out, well, let's use the super secret exit there.
Super secret exit. You mean behind here?
Nobody's ever touched the drape before.
There's a lot of things that you haven't seen yet. That's okay.
Thank you again.